Hi, my name's Alex, and as a human geneticist, I'm fascinated by the diversity within our DNA. If I was to sequence your genome and compare it to mine, there'd be 5 million differences. It's these differences, or genetic variants, that make each of us unique. My research focuses on how genetic variants influence our health, particularly regarding blood clotting disorders. Think of a time you cut your finger. Annoying, yes, but for most of us it's a minor inconvenience, thanks to hemostasis, our body's natural ability to stop bleeding. An essential part of this process is the conversion of fibrinogen to fibrin. Fibrin forms a mesh which allows us to form a stable blood clot. Yet, some individuals have genetic variants which cause them to have a low level of the fibrinogen protein, or a dysfunctional one. These individuals have a congenital fibrin disorder. However, individuals with the same disease-causing variant can have a wide range of symptoms. For instance, an individual with disease variant A could experience bleeding from severe to mild and/or thrombosis, as well as being asymptomatic. Remarkably, family members with the same disease-causing variant can have different symptoms. This variability is a significant challenge in our field, as it complicates our ability to be able to predict the symptoms a patient might develop, which in turn makes prescribing their treatment difficult. My research looks into why we have this variability in these patients. My hypothesis is that the presence or severity of a patient's symptoms are not only solely determined by the disease-causing variant. Instead, they may also be shaped by additional genetic variants. Remember the 5 million other genetic variants we all had? Well, these could also affect the hemostatic balance and shape the patient's clinical symptoms, especially when fibrinogen, one of the leading proteins, is compromised. To test this hypothesis, we're conducting an extensive review of the genetic variants in all the genes of hundreds of congenital fibrin disorder patients. We do this by sequencing their DNA. This allows us to identify the disease-causing variant, but also potential genetic modifiers. By analysing the presence of these genetic modifiers alongside clinical data from patient records, we've been able to make associations that support our hypothesis. For instance, an individual with disease variant A and genetic modifier 1 could have a severe bleeding phenotype. The ultimate goal of my PhD project is to use this massive amount of genetic information coupled with the patient data to produce a predictive model. Such a model could be used to predict the symptoms a patient may develop in the future based on their genetic profile. This would make a significant difference to the treatment plans of these patients, as well as paves the way for personalised medical care.